Hi everyone, my name is Nikki and I run the Genomics Core here at the UNT Health Science Center. I wanted to start a series of videos demonstrating some of the services we offer so our customers can get a better look into what they're actually purchasing. So today I'm briefly going over the TapeStation workflow for nucleic acid fragment analysis via automated electrophoresis. This method is standard with our typical QC workflow with any library prep or sequencing service, but we also offer it as a standalone service. So let's jump in. For this video, I'm using the high sensitivity D5000 screen tape and reagents, which has a sensitivity of 5 picograms per microliter and a sizing range from about 100 to 5,000 base pairs. However, we do offer all TapeStation applications, including high sensitivity and broad range RNA, DNA fragment, as well as DNA genomic assays. So to give you an overview of the actual instrument, this TapeStation can run anywhere from 1 to 96 samples at a time. Each screen tape can run up to 16 samples, but we can load multiple tapes at a time and the instrument will just switch them out automatically. So here's a view of the software. It's actually super intuitive. Since we're using under 15 samples today, we're just going to select the positions for the tubes rather than the plate for our samples. The layout in the software is exactly how the layout is on the instrument. Once I select the positions, then I type in the names, and that's it for instrument prep. To get these reagents ready, I already had them sitting at room temperature prior to this video for about 30 minutes, and I just do a quick vortex and spin to make sure that everything is homogeneous before going forward with the assay. So for loading or prepping the actual samples, we just use 2 microliters of the sample buffer that comes in the kit, as well as 2 microliters of the actual sample or ladder. The amount of ladder and buffer you use will depend on how many samples you're running, since the instrument will need to run a new ladder for each tape it goes through. The buffer is essentially a ready mix with the gel, lower marker, upper marker, and other reagents needed for the electrophoresis. Once everything is loaded, we do a vortex for 60 seconds and then do a spin down to make sure all of this solution is at the bottom of the tubes with no bubbles. Sometimes getting these lids off are kind of tricky since you want to make sure you don't get any splashback on those reactions. I definitely have had my fair share of spinning down, opening the tubes, and then having to spin down again over and over because the reagents splattered on the wall of the tube. Patience is key. Anyways, the instrument is set up already and we're now done prepping our samples. Now all we have to do is load the samples into the instrument in the same positions we selected on the software. Next, all we do is hit start. The whole process is actually super fast, and with only 3 samples and a ladder, we can get our results in less than 5 minutes once the run is started. So now that everything is done, we can see our fragment size distributions and relative intensity among samples. It is important to note that tape stations and bioanalyzers aren't super accurate in terms of quantifying the amount of sample, so we definitely cube it as well in order to get better molarity estimates. Oh yeah, so I've teamed up with Illumina to host a quarterly happy hour for anyone interested in networking with local genomics cores in the DFW area. Definitely contact me if you're interested in this, and I will get you on the mailing list to receive invites to these events. Well, thanks for watching. Till next time.